Good afternoon, everyone. This is Peter Cooper from the NCBI. We've got two webinars today. One is a practical guide to NCBI BLAST, and you're seeing sort of a cover slide for that there. The other one begins about an hour and 15 minutes later, and that's a webinar on human variation resources and medical genetics resources. These were originally requested by Des Moines University, so I just wanted to give a shout out to them. They should be listening in the classroom out there. We'll record both webinars. They will be available on our YouTube channel in the webinars playlist. Take a couple of weeks for them to get up there. The materials for these webinars are on the FTP site, and there's a compressed link that will take you there. It's going to have the slides in there, as well as the demos that I'm going to do after I give the slideshow. This is a shortened version of a larger workshop that we give that lasts a couple of hours. So if you have any questions about this, my email address and contact information is on the front. What we're going to do today is talk a little bit about the basics of using BLAST, the reasons people use it, some of the statistical information that BLAST gives you, the scoring systems, what the search programs are, and talk about some other alignment services that are not BLAST. We offer them on our website. And then I'm going to go over to the web browser and do some live searches. So BLAST is probably the most widely used sequence similarity search tool in the world. What it does is it finds high-scoring local alignments between two sequences. They could be protein sequences or DNA sequences. BLAST includes a model of score distributions for random local alignments, and because of that, it can provide some statistical information about how different your alignments are from chance. So BLAST tells you then about non-chance similarities between biological sequences. That's interesting because if the similarities are not due to chance, then they must be due to something else. A couple of things that could be. The most interesting one from the point of view of the original purpose of BLAST is homology, that these two sequences are descended from a common ancestor. In most cases these days, people are using BLAST for simple identification. That includes things like annotating things like genomes or seeing what kinds of problems there are with the alignments of things to a genome. All BLAST searches begin with a sequence, either a protein or a nucleotide sequence. That could be either one that you determined or it could be one from the database. Let's talk for a minute about BLAST statistics. The most important statistic you get back from BLAST is something called the expect value or the expectation value. It ranges from essentially 0 up to the size of the database. It's the number of alignments that you would expect by chance with a particular score or a greater score. So for example, if we have a 5 base pair sequence like this one, Elvis, that has an expect value of 48,000. We would expect to see 48,000 hits that good or better in a database search by chance. So we don't know anything about that particular hit. The one below it, though, has an e-value of, that's read as 7 times 10 to the minus 18. That means I wouldn't expect to see any hits that are that good by chance. So that tells you about your alignments are different than chance, and if they're different than chance, they're due to something else. A real important point from this slide is that the e-value depends directly on the size of the search space, the size of the database. You want to search the smallest database that's likely to contain the sequence of interest. We'll come back to that point when we talk about limiting your database. So BLAST uses two schools of thought in terms of scoring things. The one that's the classic kind of BLAST scoring system is called a position-independent scoring system. That means that the same substitution in your alignment gets the same score at any position in that alignment. The model that really assumes is that all positions in a sequence are equally likely to change. That's not a realistic model for the way proteins or DNA sequences evolve. Nevertheless, this is used by ordinary BLAST. BLAST-P uses the BLOSSOM62, and it includes the concept of conservative substitutions. Nucleotide searches are less sophisticated. They use an identity matrix. The other kind of BLAST, which we're not really going to have time to talk about today, but we will see the results of, are position-dependent scoring matrices. In those kinds of scoring matrices, the substitution score depends on the position in the protein or in the alignment. This means, of course, that some positions are more important and less likely to change than others. And that's a realistic model for the way proteins and other biological sequences evolve. The programs that do this, they're SciBLAST, DeltaBLAST. Those search a database with a position-specific score matrix or a PSSM. Reverse SciBLAST searches a database of PSSMs and identifies conserved domains. And that's the search that we're going to see today that uses these position-specific scoring systems. All BLAST programs also include some kind of a penalty that allows them to incorporate gaps in the alignment. Okay, so let's talk for a minute about what these BLAST search programs are, what they're called. So the nucleotide search programs that you're going to see on the web are BLASTN and MegaBLAST. BLASTN is a traditional BLAST algorithm. It's the most sensitive kind of nucleotide search. MegaBLAST, by the way, is a default algorithm. This is the best program for simple identification, same species annotation. You just have to remember that that's the default algorithm on the BLAST pages when you do searches. You may sometimes need to change it to the more sensitive BLASTN. There's sort of an intermediate algorithm that's not used very much called discontinuous MegaBLAST. It's also more sensitive than MegaBLAST. And then here are the protein search programs, and today we're really going to only focus on the position-independent scoring ones. There's BLAST-P, which is a protein-protein search and alignment program. And then there are these translating searches. These are useful for unannotated protein coding regions, and we'll do one of these today. There's BLAST-X, which translates your query sequence and searches a protein database. T-BLAST-N, which translates the database and searches it with a protein. And T-BLAST-X, which translates both the query and the database. But all of these things are searches at the protein level. So when you search BLAST at NCBI, how do you get here? Certainly you can get there from our homepage. There's a link right there at the NCBI homepage to BLAST. There's also the most common way to do it for most people is simply to type NCBI BLAST in Google, and that will take you right to the BLAST homepage. This is the current look of the BLAST homepage, and by the way, this is going to be changing fairly soon. The furniture is going to be rearranged. There's no real difference in function. Basically, this is divided into several sections that are useful in terms of what they do. One is a way to get access to assembled genomes, and I'll show you that in more detail in a few minutes, that you can pick any genome that you want from there and find the most complete set of data. Then the center part of the page is basically the core of the BLAST searches, and we're going to be focused on that today. This is what we're calling basic BLAST. Those links take you to pages that are preloaded to do those kinds of searches. And then finally at the bottom, there's sort of a miscellaneous set of search programs or alignment tools that are related to BLAST but are not necessarily BLAST. And we'll visit some of those today. When you're using BLAST, you need to have something to search with, and that's your query sequence. I just want to point out a few aspects of this that still confuse some people. The BLAST search programs on the web will take FASTA formatted sequences, like those shown in the upper left of this slide. They will also take accession numbers, NCBI accession numbers. We'll pull those from the database and do a search with them. You can also run BLAST directly from the entree pages, nucleotide and protein. We'll do a couple of examples like that today. The other point to take away from this slide is you can use multiple queries in a single search. Most people know that, but occasionally we run into somebody that thinks you can only do one sequence at a time. Each sequence will be searched separately as a BLAST search if you do this. I want to talk for a minute about something that's a little bit odd if you think about it. One thing you can do at NCBI, which is useful, is to compare your own sequences without doing a database search at all. I just want to point out the two options for doing that at this point in the talk. One is BLAST two sequences. And so any BLAST page at NCBI, when you go to the BLAST form, there's a little checkbox that says align two or more sequences. And if you check that box, another box will open up, another text field will open up on the form, and you can enter sequence in there. You can enter many sequences in there, so you can do your own little database search against your own customized database if you want. You can also access this under that specialized BLAST section at the bottom of the page. One thing we know from talking to people at the help desk here is that many times when people are doing BLAST2 sequences, what they really want to do is a global sequence alignment. We do have that available in the specialized BLAST section. It's not BLAST at all. This is an algorithm called Needleman-Wunsch, and it allows you to compare the entire lengths of two sequences. This is a global alignment tool. It doesn't provide any meaningful statistics about whether this is a chance alignment or not. It will just align anything to anything, and it will include all the residues of your particular sequence. So if you want to do global alignment, if you're interested in knowing what the percent identity is between two proteins, this is the tool that you would have to use to do that. Okay, so now those were searches independent of our databases. Let's talk a little bit about the BLAST databases, and this is sort of a complicated part of our system that you need to sort of understand what's going on there. Some of it's a bit chaotic. The protein databases, which you're searching using either BLAST-P or BLAST-X, are fairly straightforward. We do have sort of a comprehensive database called NR. This is a non-redundant database. It contains the majority of the protein sequences that people are interested in at NCBI. It also has available useful subsets on the database pull-down list, RefSeq, SwissProt, PDB. Just keep in mind there are some sequences that are not part of the protein NR. US and European and Asian patent sequences that we get are not in there. They're in a separate database. The proteins that are coming from metagenomic samples, from sort of ecological genome things, those are not in there. And also the proteins from next-gen assemblies. So these are transcriptome shotgun assembly sequences. This is a growing set of data, in particular for the nucleotide side, but there are some TSA proteins as well. Those are not part of NR. This is what the nucleotide search page database pull-down list looks like. And it's quite a bit more complex. And I've got you can certainly, anytime you go to a BLAST page, you can click on one of those question marks to get information about what sequences are included. This is a slide that has some more details about that. A couple things to keep in mind about the nucleotide databases that make them different than the protein. The main one is that the default database that we call NR, I like to refer to as NT, which is what we call it on the FTP site. This is not a comprehensive database. It contains the traditional GenBank sequences, things that are not bulk sequences, and our NCBI RefSeq RNA sequences. That's actually a very small set of data compared to everything else we have at NCBI. It's a useful set, but it's a smaller set. Some subsets of that, which are cleaner, are the RefSeq RNA database. There's also a 16S RNA database as well that you can search. So what's not in NR is the majority of the nucleotide data. That includes all the bulk sequences, the RefSeq genomic sequences, which include our chromosome records and our various sizes of assemblies there. The patents are not in there, and some other large sets of data, including the whole genome shotgun sequences, the transcriptome shotgun assemblies, and SRA data, which is really the largest set of data at NCBI. It's so large that there's no way to actually search it as a single entity. We'll talk a little bit about that when we do a demo. Another set of databases that are really separate BLAST pages, if you will, are available through that device at the top of the BLAST homepage, which lets you search genome assembly databases. Basically, this is a way of getting you a BLAST page that has the most completely assembled genome for that particular species. So you can type an organism name in there, and then you can link directly to it, and that will take you to a BLAST page set up to run that search. Now, I mentioned this earlier in the talk, that the most important thing you can do when you're using BLAST is to search the smallest database that's likely to contain the sequence of interest. And that's because the database gets larger and larger as it gets larger and larger. It's harder and harder to discern the signal from the noise that's in there. And that has to do partly with the way the expect value scales with the size of the database. So there's some useful things that you can do here. You can use one of the organism limits. You can type the name of an organism or a group of organisms. You can even exclude groups of organisms that you don't want to see. So here's an example, getting all the bacterial sequences without the order Enterobacteriales in there. You can get rid of things like model sequences or uncultured sequences if you're working with bacteria. You could even specify things like a molecular weight range. So any entree query that works in the protein or nucleotide database will also work on this page. Okay, a couple of things to finish up here, and then we'll pause and see if there are any questions. One of the things that, as a person who manages BLAST Help or sits on the BLAST Help desk here, it's very important to me and to all the other people who sort of support BLAST, is that you understand that there is an identifier for your search, and that's called the request identifier. If you look at your BLAST results, it's at the top. It says RID, which might not be clear what that stands for. But that identifier is a unique identifier for your results. So if you have a problem with a BLAST, you can write to us and give us that identifier. If you click on that link, it will give you a URL like the one in the middle of this slide. And you can just paste that in a web browser and get your results back, or you can send it to somebody you know that you want to show them your results, or you can send it to us. We will keep your results on the servers at NCBI for about 36 hours. You can see them through the recent results link that's on the BLAST page. They'll also show up in your My NCBI. It doesn't make them last any longer. They still last for 36 hours. So keep this in mind, and we'll show you that, managing that a little bit later on today when we do search. So be sure to send us an RID if you have a question about a particular search. We can look up your results and see exactly what the settings were, and we can figure out if there's a bug or if there's something we can help you with to make the search work more efficiently. The other thing I want to mention is that BLAST offers a number of download options. This is actually an older screenshot. We've added a couple of more structured formats here. Just be aware that they're here. These are the kinds of things that you'll want if you need to save BLAST results or to save huge sets of results to try to parse out information from them, because they're structured formats that you can parse with a script, or you can use some of the utilities that come with BLAST to redisplay them. And the hit table is particularly popular even with people who don't script, because that can be loaded into Excel, the CSV version of it in particular. Okay, so let me talk about some of the specialized BLAST services, then we'll stop for questions. Bonnie's looking at me because I said we'd stop for questions next. And these are ones we're going to demonstrate in a few minutes. Primer BLAST is our primer designer and specificity checker. It takes advantage of free software, Primer 3, to design the primers, and it uses annotation on our sequences if you want to design primers that do things like span exon boundaries and things like that. And it uses BLAST to make sure your primers are specific. BLAST is a tool that's very specialized, and I won't demonstrate that today. We have done webinars on this particular topic. This is a way of clustering sequences and finding a taxonomic placement for things like 16S sequences. We use it internally in our taxonomy group to help identify things. Two special protein services that I will demonstrate today are Cobalt, which is our multiple alignment tool. Cobalt stands for Constraint-Based Alignment Tool. It does a protein global multiple alignment. Just like Needleman Wunsch, a global alignment tool like this requires that you input sequences that you know are related to each other, because otherwise you just get a mess. The beauty part about Cobalt is it lets you take the output from a BLAST search and feed it into Cobalt. So you already know those sequences are related, and you can run it as an extension to your BLAST search. And then I'll give you a quick demo of a new tool that's kind of something we're sort of trying out as a rapid protein identification tool. It's called SmartBLAST. It uses a very rapid approach to searching that uses K-mer content of sequences to find matches. It's very quick, and it might replace some of our internal mechanisms for neighboring things like proteins to give you a live search, say, if you wanted to find related proteins on the web. And it uses Cobalt to produce a multiple alignment and a protein tree. Okay, so now I'll just mention that there is a help link on the BLAST pages, and this has lots of good information, including links to their handbooks chapters, the help documents, and the YouTube channel, which has a lot of BLAST tutorials. In fact, this will go on that. In addition to the webinars playlist, they'll go on the BLAST tutorials playlist on our YouTube channel.
3 questions.
So Bonnie says we have 3 questions.
The first question is what's the limit for multiple sequences in the query data set?
So that's a question that we get often at BLAST Help in particular. You're allowed, the way it works is you're allowed 1 hour of CPU time. So that's processing time. It's not real time. That could be just a few minutes of real time depending upon how many processors your search runs on. So there is no fixed limit based on number of sequences, number of residues, but you will run up against it fairly quickly if you use large numbers of sequences that have a lot of hits in the database. So I'm afraid I can't give you a concrete answer. If people write in with proteins, I would say, no more than 100 at a time. For nucleotide sequences, depends on the length, because that can vary a lot. But if you're trying to do something like search with chromosome 1 against the NT database, Bonnie's laughing, but this happens all the time. Don't do that. It's not going to happen for you.
Because chromosome 1 is how long, Peter?
I don't remember. It's big.
Okay.
um,
The largest human chromosome, so.
There are lots of things that you can do to sort of ameliorate that problem. But if you have a need to blast hundreds, thousands of sequences, you might need to think about some other options. And I can point some of those out to you, and they're available on the Help tab under the Developer options. Okay, do we have another one?
The question was, is the protein reference database not included on NR? And I wanted to make sure that the person meant the BLAST-P protein NR database, and I didn't get a clarification.
Well, the answer is really the same on both. So the RefSeq, well, as long as we're talking, well, let's address them separately. On the protein side, RefSeq proteins are included in NR. That's easy. The nucleotide side, the messenger RNA, the transcript sequences are included in the NT-NR database, the nucleotide default database. The larger reference sequences, the assemblies like chromosomes and contigs and things like that, those genomic sequences are not included in NT-NR. And there was a third question?
The person says that the BLAST results are missing right away after they did the blast, and they can't find them.
I'm not sure I understand the question. So
I know I didn't either completely, but I hoped that you on the blast help might have seen that before.
Yeah, no, I don't know what that person means by they can't find them. I don't know. Maybe you can rephrase that and we'll come back to it later. So I'd like to stop here and go on to do a few live demos. What I want to do is to do a few searches, and we're going to, I've got actually a document on the FTP site that goes over what we're planning to do in the live searches. So I'm going to do a couple of things with a mammalian protein called creatine kinase B, the brain type creatine kinase. We're going to do some blast searches with that. Then we're going to do a translating search against a fish TSA database to find the corresponding nucleotide sequence for a protein. We'll use a different protein for that. We'll use glycine dehydrogenase. And then actually, I will give a quick demo of SmartBlast using an open reading frame that I got from that fish sequence. And then we're going to do two other searches. One is to show you some things about the nucleotide system by searching the human genome with a transcript from Macaca fascicularis. And we're going to design some primers using PrimerBlast. There is another example in here on using SRA, but we won't have time to do that one today, I'm pretty sure, because I want to spend some time on those first several. Okay, so what I'm going to do is we're going to start actually not in BLAST, but we're going to start in the protein database. And I'm going to set some searches up for you. Notice that I also have, I mentioned those BLAST RIDs. I have those in this document, and they're stable for a while anyway. We have the ability to preserve these at NCBI. At some point, they will go stale because the underlying things change and they don't work anymore. But I can use those to retrieve my results to save us some time, in particular for the first search, which I'll set up, that I'll show you by just retrieving the results, why it's important to limit your database. So I'm going to go over here to the NCBI homepage, and I'm going to change my database to protein. I'm just going to retrieve a sequence that I happen to know the accession number for. This is a creatine kinase from human. I know the accession number. This is a reference sequence accession number. I'm going to retrieve a human reference sequence. My goal ultimately with this search is to try to collect a set of sequences to do a multiple sequence alignment with them. And I'm going to try to collect mammalian creatine kinases. So notice that from any protein sequence like this, I can click the Run Blast link here. And really all that did for me was it just loaded the BLAST page for me with the accession number in the query box there. And I'll talk about some of the settings on the page. I normally have slides for that, but I thought I'd just do it live because I think we can do things a little bit more expediently that way. One of the first things you need to do when you come here is to figure out what database you're searching. And here's the pull down list. Now I'm going to leave it set to NR for a moment. I'm actually not going to run that search for you. I'm going to retrieve the results because that's going to take a while. And this is a good example just to illustrate for you the problem that you're going to run into now with a size of the protein database being so large and so heavily weighted in sort of two areas. One of those areas is the vertebrate proteins, and the other area that's heavily weighted in is bacterial proteins. And that has to do with the efforts to sequence and annotate things. So we'll come back to limiting this in a few minutes. There also is a set of, there are some settings below this fold down here that says that it's called algorithm parameters. I want to just show you that briefly because there are some things down there that I think you may need to change sometimes. One of the important things here is there are two parameters that govern your output. One of them is the maximum target sequences. So no matter what else I do, BLAST will not show me more than 100 sequences. It's a little bit worse than that, though. What it means is BLAST will not collect more than 100 sequences, and BLAST is a two-stage algorithm. So if you have this set too low, you can wind up missing some important things. So you can increase this. In fact, the search that I'm going to show you, I set it to 5,000. And in fact, that wasn't enough, as you'll see in a minute. You also will probably want to adjust your expect threshold ordinarily. Notice that it's set to 10. That means that the worst score that I'm going to show you, I would expect to see 10 hits that are that good or better by chance. That's not something that's terribly interesting to you, so you can set this to some other value. There are lots of good arbitrary values to set here. One that's quite common for protein searches is this one, 1 times 10 to the minus 6. I think that's a useful one to use there. You could set it to 1 times 10 to the minus 3. But that means that at least there's some kind of possibility that that's not due to chance. The other thing to notice that I want to point out here now that's a recent change in BLAST, for protein, one of the shortcuts that BLAST uses is it doesn't find or try to extend every match. It finds matches of a certain size and then starts to extend them. The default once was 3 for protein searches. It's now 6. That makes the searches faster, but just be aware, if you come here and you find that you don't get exactly the same results that you got, say, last year at this time, it might be because of the word size. It's going to affect sort of marginal hits in many cases. So those are the things I wanted to show you here for the protein pages. And I could run this. It may take a while to run. So what I'm going to do is go back over here and retrieve my RID. And so I've got one here that I ran that has this RID here. There's a complete URL there. I can also just show you that I can get this from my recent results. I'll just copy that. I'll go over here to this link that says recent results, and that's available on any of the BLAST pages. And I'll paste this in here. Now, I actually did do some filtering on this, so I eliminated some of the model organism searches, and I'll come back to that in a minute when we go back and show you this when I'll resubmit it, and we can see all those settings. The main point in showing you this is that, first of all, we did run conserved domain search on this. It has a phosphagen kinase conserved domain. It's a creatine kinase, so that's what I would expect. If I didn't know what this protein was, it would give me some ideas of what the function of this protein is. I've maxed out my display here for the graphical overview. It holds 100. I can change that if I want to. And then down here, I've got my what we call the BLAST descriptions. These are the hits essentially, and they're sorted for me by e-value. Notice I have all these e-values of 0. They're not really 0. They're just a very small number. And I'm trying to reach my cutoff, which remember was set to, I think when I ran the search, I probably left it at 10. I might need to reformat this because I don't have everything. So let's make sure that I get all my results back. What I can do here is go back here to the formatting options. We'll use this more than once today. It's still set to 100 descriptions, but notice that I can get up to 5,000, which is what I originally requested. And so you can see what happens very quickly is that I get a tremendous number of hits that are from all kinds of life forms. I should be able to take this all the way back to the bacteria because it has a conserved domain. So here I'm back into some things that are insects. that have arginine kinases in them. But this is an overwhelming amount of output. And the main point that I wanted to make to you with this search is that there's no reason for you to do this because you probably don't need all this. You probably want to do something much simpler and restrict to a particular set of organisms. So this just goes on and on and on, 5,000 hits. And by the time I get to the bottom of my descriptions, I still haven't reached my e-value cutoff. And that means that I'm missing significant hits. Maybe that significant hit is one that I'm interested in. So the main take home from this is to make sure you're limiting your database to something that you're interested in. And this one I already did limit a little bit. Let me show you what I did. So if I have a result like this and I want to resubmit it, I can click this link that says edit and resubmit. And this will take me back to the page that shows me exactly what I did. So I did a search with the creatine kinase. I did this yesterday. I searched in R, and I did get rid of some kinds of sequences. I got rid of models, and I got rid of uncultured environmental sample sequences. I did ask for 5,000 hits. I didn't restrict my e-value, and I never reached it. I had an e-value set here for 10, but I never reached that at all. I never even got to an e-value that wasn't significant, because even with 5,000 hits, I didn't find all the significant matches. This down here was on by default. This is a filter for the kinds of sequences that violate blast statistics. This is low complexity reason. So if I want to fix this to make it a little bit more manageable as a search, probably the best thing that I can do is to run it against a particular group of organisms. So if I'm interested in making a little phylogenetic tree or a protein tree, and I would want to collect sequences for my group of organisms, so I'm going to collect mammals, which is a much smaller set of data. And let's look for a better controlled set of data, and let's do the RefSeq proteins database. So I've now made the database smaller, and I've made this RefSeq protein database even smaller by doing two things: restricting by organism and getting rid of the model sequences that our pipeline is producing. And the other thing I might want to do is to go down here and to change my expect threshold like we did earlier. So I can make it to something fairly significant. And these are just arbitrary cutoffs. You can go back and change them if you find that you're not getting sequences that you want or you're getting things that you don't want. So I will try to run this one live. Let's see how long it takes. So immediately I have my conserved domain result. That's partly because this sequence is in the database. We already know what conserved domains are on it. Remember, that's a position-specific kind of search. And if I think this is taking too long, I can go back over here because I have run this and get my request ID out of my little document. And if you want to come back and retrieve these, they will work for a while, probably several months. They won't work next year. Where we are here. Well, I didn't need to do that because it's done. So now instead of maxing out everything, I have 32 blast hits. And you know, if I look at my graphical overview here, you can see there's sort of two kinds of hits. There are these longer ones. This is the one that we started with. These are the cytosolic isoforms or genes for creatine kinase. And notice that there are some that are going to the mitochondria, and you can probably guess that the reason that they don't align is because there is a leader peptide at the beginning here, a signal peptide that tells the cell to send that to the mitochondria. Looking at my output here, I can see the organisms in the list. My e-value was set pretty stringently, but these are all very similar proteins, so I got them here. That looks pretty good. If I wanted to look at an alignment just to see how BLAST does an alignment, let me look at this one from pig, say. If I go here for the pig U-type, it jumps me down to the alignment, and you can see the way BLAST shows a protein alignment. Notice this is a local alignment. So the first 11 residues of my query didn't align, and the first 44, of my subject sequence didn't align with the query. And then you can see how the center line is sort of reflecting the scoring system. These plus signs represent positive scores for substitutions in the underlying BLOSSOM 62 matrix. The blank spaces means that that's a negative or a 0 score in that matrix, and the identities, of course, are given a letter there. I don't see any gaps in this particular alignment, but if there had been some, it'd be rendered it. Oh yes, here's one right here. There's a gap right there. So BLAST inserted a gap there that was cheaper than aligning the residues incorrectly at that position. So I have an ad here for SmartBlast, and we can do that next with a different protein after we do another kind of protein search. Does anybody, let's pause here for a minute, Bonnie, and see if there are any particular questions about this one.
Not about this, the clarification from the missing BLAST result that I wanted you to address was sometimes the network crashes during the, so I just wanted to see if you knew or could address. So when you make the BLAST request, it gets to the NCBI servers. The NCBI servers can run it, and the results should be in the recent results window. But when if there is a network crash, at what part of the process would that prevent the BLAST?
I don't know.
Okay.
If the person has a particular issue, they should write to us.
Okay.
And we can help them solve the problem.
Right.
Because I can't completely understand what the problem is from the question. But write to BLAST Help or write to me, and with enough details about exactly what happened, what your search was. If you got to the point of getting an RID, send that to us too.
Right. There is another question about, is 5,000 the optimal value for protein, or does it depend on the size of the query sequences?
No, you mean 5,000, it depends on what you're trying to do. It depends on what you're trying to do. The main thing is to make sure that that isn't limiting your results, that the expect value cutoff is limiting your results. Okay, so I want to do a different kind of example. We're going to use another protein search just to show you what a translating search is useful for. And I'm going to go back up here to the BLAST homepage. And notice that I can choose from, here I'm going to take a protein sequence. I'm going to take a highly conserved protein, and I'm going to try to find it in one of these transcriptome shotgun assemblies. These are assemblies from NextGen RNA-seq data for organisms that we basically have no other kinds of data for. Many of them have no protein data to speak of at NCBI. But those assemblies of their transcripts, you can identify the corresponding regions. So let me just show you that real quickly. I'm going to go to tBLASTN. And we're going to use as a query sequence one of the ones that's in my output over here. And this is glycine dehydrogenase. So again, the query sequence, that's not that hard to do. But notice that my databases here are different. So I'm using a protein query, but my databases are nucleotide databases. This is a growing set of data called transcriptome shotgun assembly. In many cases, those assemblies are also represented in SRA, so you could potentially search SRA as long as you knew what the experiments were that you needed to search. The transcriptome shotgun assembly database is set up the same way that WGS is on BLAST. So when I choose that database, notice that I have to do something else. So choosing an organism, for example, is not optional. I have to choose one. So the organism that I'm going to choose here striped bass. Okay, so that's a organism that's near and dear to a lot of people's hearts around this part of the world in the Chesapeake Bay. So there's a transcriptome shotgun assembly for that organism. If I want to, I can change my expect threshold just as a matter of course to be something a little bit more significant. And I'll leave those settings the way they are, and let's see if this is fast. These searches are a little more burdensome than doing an ordinary BLAST-P search, because what BLAST is doing is it's translating the database in all 6 reading frames on the fly to give you a protein. So I have a very nice hit here, of some other sort of minor hits. And if I click down here, here's my match. This is the translation of my subject sequence. And it's a pretty decent match to give me the pretty good idea that this is a homolog of that protein. When I go here, this doesn't take me to the nucleotide database. These are stored in a separate system. This is going to our WGS browser, which also contains the transcriptome shotgun assemblies. I'll go ahead and open that in a new tab. So what I can do there is to get the FASTA sequence, but this exists basically only here. I can't get this out of the normal nucleotide database. There's a master record for this here. I could download the entire set if I wanted to. Now what I did actually was I translated that, and I got a little protein sequence. And this would be a common use case for SmartBlast. That I have a protein that I've generated from some kind of project like this, and I want to identify it. So let's see what happens when I use SmartBlast to do that. So I'm going to go back over here, and this is my open reading frame that I got from that sequence. And I'll show you a couple of aspects of SmartBlast that are kind of useful. One is the rapid identification. The other is that it does give me a little bit increased look-back time because the database is smaller, and I don't run into that problem of being limited by the number of proteins that I can collect. So I'm going to go back here to the BLAST homepage. I'm going to retrieve the SmartBlast link here. I'm going to paste that protein sequence in. So I search with a bony fish sequence, which is labeled as unknown, and notice that it places it in this nice little protein tree for me. And if I wanted to know what this protein was, then I have no question that this is a glycine dehydrogenase decarboxylating mitochondrial form. So I have other fishes there, the yellow croaker, the damselfish, the guppy, the zebrafish. Notice the hits are in 2 different colors. There is a reference database, or what we call a landmark database, of proteomes from well-studied organisms. The house mouse and zebrafish are 2 of those. If you go to the Help tab, it defines what all the other organisms are that are in that database. In addition, it gives me hits from the best hits from the NR database. So that's where this large yellow croaker, the damselfish, and the guppy sequence come from. So that's very good, and it was very fast at identifying that. The other thing that's kind of useful about this, so here are my best hits, top 5. But these additional hits are interesting because it let me look back. Because the number of organisms in that reference database are limited, I can see much further back than I could in a search against NR. So here are some bacteria, Thermatoga maritima. In fact, if I wanted to find Escherichia coli, I could just do a find in page to make it easy. So here's my match to E. coli, which I defy you to find that easily on a search against NR, because what happens is you're going to have to get many, many thousands of hits to see that one. It's still a significant match. It's only 33% identical to the protein that I started with. So I'm going to change gears and do one thing with BLAST to just show you some of the formatting options that are useful. All right, so let me go over here to the BLAST page again. Actually, what I think I'll do is I'll start with a nucleotide database, which is one way of doing this search that I'm going to do now. I'm going to go back here, and I'm going to do a search using a sequence that's actually got a problem with it. We can see that problem very easily by using one of the formatting options in BLAST. So I'm going to go ahead and copy that. That's a nucleotide sequence. So this one has sort of a funny definition line. It was some kind of a high throughput cDNA sequencing project, It says it's similar to CDC20, but there actually is a problem with this sequence. This is from a monkey. I'm going to click the Run Blast button here, and I'm going to throw this into the standard nucleotide blast page, but there's a shortcut here that's pretty handy. It'll let me search the human genome. So I'm going to do that. Now, this is my first nucleotide search today. Notice that this is set to megablast. That means that it's not very sensitive, and I won't have the kinds of problems that I have with protein where I'm looking back really far and seeing lots and lots of hits. This is an example really of an identification search and a search that looks at annotation problems. We'll go ahead and run that. That was very fast because we have an index search of the human genome. We actually have two sets of data with two genomes. We have the transcripts. I hit the corresponding CDC20 transcript. And then we have hits to two different genome assemblies. We'll focus on the primary assembly. So if we look at the alignment to CDC20, I can go here and take a look at that. There's some mismatches at the beginning. Mismatches, but it's a very close alignment. One of the things I want you to notice is there's a little gap right here, and that's near the 3' end of the alignment. That might not be a big problem if it's in the UTR, but what if it's in the coding region? It could cause a frame shift. So we could see, if we could add the coding regions onto this, then we can see what's happened there. And so that's the main thing I came here to show you. If I go to the formatting options, I can add the CDS feature, which will pull the coding region features, the mRNA translations from the nucleotide sequence database. And I can also render this in a way that's going to give me some indication of where there are differences. It's kind of hard to see where the mismatches are and where the gaps are and things like that. Let me reformat that. So here's my N-terminal methionine. There are some changes in the coding region here. They're kinds of subtle things, serine for alanine. Some of them are silent substitutions here. But here I have the problem. So it looks like the gap that was inserted here, it looks like there must have been a sequencing error probably in that macaque sequence. And so it threw a frame shift in here. Beginning here, there's a different reading frame translation of the sequence. And so this kind of formatting option is very useful for seeing those kinds of things. The other thing I want you to see here is the way that you can look at hits to the genome. There is a hit to a pseudogene. I won't go into that right now. You can look at that later if you want to. I'm just going to look at the main hit, which is on chromosome 1. We're down in the alignments here, and so we're sorting this by E-value. We can sort this not by E-value, but by query start position. That will give us basically the exons in order. Now, they didn't line up exactly right because BLAST doesn't know about splice junctions. The other thing that I can do here that's very useful to be able to do is to just display this in the graphical sequence viewer, and so we can see what's going on. I'm going to display this here. My BLAST hits are going to be loaded in the graphical sequence viewer. In this case, it's sort of inverted my BLAST hits, so it's showing me this subject sequence with the query sequence aligned to it this way. And so you can quite clearly see the exon-intron structure here with the mismatches highlighted. I'm going to use the graphical sequence viewer just to zoom into this 5' end. And you'll notice that I missed the first exon. One of the things you can do as an exercise or to convince yourself that it's true, if I use a BLAST-N, which is a more sensitive kind of search, that I will make that first exon, I will be able to align to it. MegaBLAST, which is what we used, uses a very large word size shortcut. It uses a word size of 28. So if there are any mismatches in that 28 nucleotide hit, it won't find a hit at all. So this first untranslated exon doesn't find a match with megablast, but it will with blast N. Okay, so I think we need to wrap up pretty soon. Why don't we pause here, Bonnie, and see if there's any questions? And if not, I can do the primer blast example fairly quickly. Do we have anything?
Well, you can set up the primer blast example while I ask this question because it feeds right into it, and that is there no RID for primer blast?
There is an RID for primer blast, and I'll show you that when we do it.
Okay.
So what I'm going to do is we're going to design primers for a particular exon of a gene. So I'm going to work with BRCA1, and I'm going to cheat and use a shortcut that's built in a lot of places. It's the gene sensor. So I'm actually going to search PubMed, and notice that there's this add for gene. Actually, I want to do this in nucleotide because it also has one. But the advantage of the one in nucleotide is that it gives me access to the genomic sequence. This is the RefSeq gene record. I can highlight sequence features here. And when I click that link, I'm able to sort of browse the features. And let me go ahead and get the exons here. So there are 23 exons of BRCA1. I'm going to pick exon 15 as the thing that I want to amplify. And this is a common kind of task that people have. They need to get primers that will amplify an exon of a gene. So notice that I can now display this exon in FASTA format. And I want to design primers that will amplify this. So what I can do is send this directly to Primer Blast as a template sequence. So this will design primers that will amplify within that exon, but if I want to, I can make it so that it starts before the beginning of the sequence and ends before, and it binds outside of the exon. So I can go ahead and copy that, move it over. Likewise, I can change the endpoint over here. Then I need to pick a background database. It's already set to the tax ID for human. I want to amplify this out of genomic DNA. So I'm going to pick RefSeq reference assembly from selected organisms. This is a good one to pick. RefSeq representative genomes also works well for human because this is the representative genome for human. I'll click the Get Primers button. It recognizes that there's a sequence that it matches chromosome 17, and that's the gene BRCA1, so that's okay. I want to say that that's right. That's what I want to find. Primer Blast can sometimes be a little slower than everything else because it's run on fewer machines. It also has a very wide open blast search at the end. And so I got 3 primer pairs that are outside of exon 15 in BRCA1. So these are a decent set of primers that will amplify that exon and not really bind within it too much. And you could actually add something to this to see that this is a region that has a lot of disease-causing mutations. And so this is a common kind of task that people who are screening things for genomic mutations would do. Now, somebody asked me about the RID for Primer Blast. You can use this job ID here to go back and get the Primer Blast results, previous ones. Yeah, so it's over here. So if you have your Primer Blast job ID, which is that long string that we had a minute ago, you can enter that in here and retrieve the result. They don't persist, any longer than ordinary blast jobs, but you can do that. Okay, so that is a wrap on this one. So those are the things that I wanted to show you. We ran over by about a minute or two. We can stay open for a couple more minutes if anybody has any questions. Okay, thanks everybody for coming, and that concludes this webinar. We will have another one beginning in about 15 minutes.